Right now, 8 independent research teams are locked in a scientific standoff over the same set of numbers, raw data beamed back from the James Webb Space Telescope, pointing at a single planet 124 light-years from Earth. The planet is real, the atmosphere is real, the water vapor, methane, and carbon dioxide detected there are confirmed by nearly everyone involved. But one specific molecule, a chemical that on Earth is made almost exclusively by living organisms, has split the astronomical community down the middle. Some researchers say the signal is there, buried in the starlight. Others say it's nothing but noise, and here's the part that should make you pause. Both groups are looking at the exact same data, same telescope, same observation windows, same raw measurements, yet depending on how you process those numbers, you can either find tentative evidence of a biosignature or find absolutely nothing at all. We're going to walk through why that happens, what it means, and what one specific number in this entire debate reveals about how close we actually are to answering the biggest question humanity has ever asked. The world at the center of this storm is cataloged as K2-18b, a name that carries no poetry but marks a very specific discovery. It was first identified by NASA's Kepler Space Telescope during its K2 extended mission, the 18th planet candidate found around its particular host star. That star is a red dwarf, cooler and dimmer than our Sun, sitting quietly in the constellation Leo. K2-18b completes one full orbit around it every 33 days, a pace that would seem breakneck by our standards, but places the planet comfortably within what astronomers call the habitable zone, the orbital band where surface temperatures could, in theory, allow liquid water to persist. In size, K2-18b occupies an awkward middle ground. It's roughly 2.6 times the radius of Earth and carries about 8.6 times Earth's mass. That makes it far too large to be a rocky super-Earth, but also far too small to be a gas giant like Jupiter or even an ice giant like Neptune. We don't have anything quite like it in our own solar system, which means we lack a local reference point for understanding what such a world might actually look like up close. Astronomers have settled on the term sub-Neptune as a placeholder category, a label that describes what the planet isn't more than what it is. This mass and size combination matters because it directly influences what kind of atmosphere such a planet could hold onto over billions of years. A world this massive has enough gravity to retain a thick hydrogen envelope, the lightest and most abundant gas in the universe. But it's not so massive that it would necessarily become a puffy, uninhabitable gas ball with no solid or liquid surface beneath. That ambiguity is part of what makes K2-18b scientifically compelling and frustratingly hard to interpret. Distance is another factor. At 124 light-years away, K2-18b is far beyond the reach of any probe we could send within our lifetimes, yet close enough that its host star is relatively bright, and the planet's transits, the moments when it passes directly in front of that star from our point of view, are detectable with enough clarity for detailed follow-up study. That combination of proximity and geometry has made K2-18b one of the most intensely scrutinized exoplanets in the sky, observed repeatedly by Hubble, by ground-based telescopes, and most recently by the James Webb Space Telescope's advanced infrared instruments. The first hints that this planet might be interesting came in 2019, when early Hubble observations suggested the presence of water vapor in its atmosphere. The announcement generated cautious optimism, but subsequent reanalysis muddied the picture. Some researchers argued the spectral signal could just as easily be methane, not water. That early back and forth set a pattern that would repeat itself louder and more publicly in the years to come. In 2023, the James Webb Space Telescope turned its infrared gaze toward K2-18b and returned data of unprecedented clarity. The results, published by a team led by astrophysicist Niku Madhusudan at the University of Cambridge, confirmed several key atmospheric components. Methane and carbon dioxide were both present, and ammonia, a molecule you'd expect to find in a hydrogen-rich atmosphere, was conspicuously scarce. This particular chemical cocktail immediately caught attention because it aligned with a theoretical model Madhusudan had previously proposed, describing a class of exoplanet he termed Hycean worlds, short for hydrogen and ocean. The idea is that such a planet could host a deep, planet-wide ocean of liquid water beneath a thick, hydrogen-dominated atmosphere. The lack of ammonia fit neatly into this picture since ammonia dissolves readily into water. If a vast ocean existed below, it would quietly absorb ammonia from the air above, explaining its near absence in the observable spectrum. That 2023 detection alone was significant. It marked the first time carbon-bearing molecules had been confidently identified in the atmosphere of a habitable zone exoplanet, but it was still just chemistry, interesting, suggestive, but not conclusive evidence of anything biological. Then came April 2025. Using Webb's mid-infrared instrument known as MIRI, Madhusudan's team announced something far bolder. They reported detecting spectral features consistent with either dimethyl sulfide, abbreviated DMS, or a closely related compound called dimethyl disulfide, abbreviated DMDS. On Earth, these molecules are produced almost exclusively by living organisms. Marine phytoplankton and certain types of algae churn them out as metabolic byproducts. They're part of the sulfur cycle in our oceans, and they contribute to the distinctive smell of coastal tidal zones. Crucially, scientists know of very few abiotic processes, non-living chemical pathways that can produce DMS or DMDS in any significant quantity. A press release from the University of Cambridge described the finding as potentially the strongest evidence yet for life beyond our solar system. News outlets around the world ran headlines suggesting we might have finally detected an alien biosignature. It was a stunning claim, and it rested on a single fragile thread of data. A set of absorption dips in the mid-infrared spectrum interpreted as matching the chemical fingerprint of DMS or DMDS. But fragile threads snap under scrutiny, and scrutiny came fast. Imagine trying to identify a single person's voice in a stadium filled with 70,000 screaming fans, and you're listening from outside the building through a wall, and the only tool you have is a microphone that picks up a narrow range of sound frequencies. You think you hear a specific phrase. You run the audio through a filter. Maybe it's there. Maybe it's just crowd noise shaped by echoes. You adjust the filter settings slightly and the phrase vanishes. Someone else uses a different microphone model and they hear nothing at all. A third person, using yet another processing method, says they hear something, but it might be a completely different phrase. That's not a perfect analogy for what happened with K2-18b, but it captures the core problem. The data Webb collected is real, but it exists at the very edge of what the instrument can reliably detect. The spectral features in question are tiny, small dips in brightness across specific wavelengths of infrared light, measured as starlight filters through an atmosphere 124 light-years away. These dips are smaller than the random fluctuations, the noise that every instrument produces simply by existing. Separating signal from noise at that scale requires statistical modeling, assumptions about how the data should be grouped and processed, and judgment calls about which molecules to test against the observed pattern. Change any one of those assumptions and the answer can change too. It's not that anyone is being dishonest or careless, it's that the measurement itself sits in a regime where certainty is not yet possible with current technology and current observation time. The universe is not handing us a clear answer. It's handing us a whisper, and we're arguing about what the whisper said. This is compounded by the fact that we've never seen a planet quite like K2-18b up close. We don't know what its surface looks like, whether it even has a surface in the traditional sense, or how deep its atmosphere runs before transitioning into something else, liquid, slush, or high-pressure exotic states of matter we can only guess at. Every interpretation of the spectrum depends on models of what we think such a planet should look like, and those models themselves are built on assumptions that have never been tested in a real sub-Neptune environment. So how does any of this detection work in the first place? How can we possibly learn anything about the air surrounding a planet we'll never visit, orbiting a star we can barely see? The method is called transmission spectroscopy, and it hinges on geometry and physics. When K2-18b passes directly between its host star and Earth, a configuration called a transit, a tiny fraction of the star's light doesn't get blocked by the planet's solid or liquid body. Instead, that light skims through the thin outer layers of the planet's atmosphere on its way toward us. As it does, molecules in that atmosphere absorb specific wavelengths of light, the exact wavelengths determined by the quantum mechanical structure of each molecule. Methane absorbs certain infrared wavelengths. Carbon dioxide absorbs others. Water vapor, ammonia, DMS, each leaves its own unique signature, a pattern of missing light. The James Webb Space Telescope's instruments are designed to capture that light with extraordinary precision across a wide range of infrared wavelengths. Webb doesn't take a picture of K2-18b's atmosphere. It records how the spectrum of light from the host star changes when the planet is in front of it compared to when it's not. The difference between those two spectra is the atmospheric fingerprint, but here's the complication. The effect is minuscule. A planet the size of K2-18b blocks only a tiny fraction of its star's light to begin with, and the additional dimming caused by atmospheric absorption is a fraction of that fraction. We're talking about variations in brightness on the order of parts per million. Detecting that requires exquisite instrument stability, long observation times, and sophisticated data processing to remove contamination from other sources, cosmic rays, detector noise, thermal fluctuations in the telescope itself, and even starlight variability from the host star. Once the raw spectrum is obtained, scientists compare it against theoretical models. These models predict what the spectrum should look like if the atmosphere contains certain molecules in certain abundances at certain temperatures and pressures. The team runs thousands of simulations, adjusting parameters, searching for the combination that best fits the observed data. This process is called retrieval analysis, and it's where much of the disagreement around K2-18b has emerged. Different teams use different retrieval codes, different assumptions about atmospheric structure, different statistical methods for evaluating fit quality, and different choices about how to bin, group together individual data points to reduce noise. All of those choices are scientifically defensible, but they don't always lead to the same answer. Here's the moment that turned K2-18b from a fascinating exoplanet into a global headline. The DMS detection, if confirmed, would represent something unprecedented. Not just interesting chemistry, not just a habitable zone planet, but a genuine potential biosignature, a molecule whose presence, in significant amounts, is extraordinarily difficult to explain without invoking biology. Madhusudan's team reported the detection at approximately 3 sigma confidence. In the language of statistics, that means there's roughly a 1 in a few hundred chance the signal they observed could have appeared purely by random noise rather than representing a real chemical signature. 3 sigma sounds reasonably convincing. In many fields, it would be enough to publish and move forward. But in high-stakes astronomy and physics, 3 sigma is considered suggestive, not conclusive. Major discoveries, like the Higgs boson particle detected at CERN in 2012, aren't announced as confirmed until they reach 5 sigma confidence, corresponding to about a 1 in a few million chance of being a statistical fluke. The April 2025 announcement didn't claim absolute proof. The researchers were careful to frame it as a tentative detection requiring follow-up, but the implications were impossible to ignore. If DMS or DMDS were truly present in K2-18b's atmosphere, and if no plausible abiotic mechanism could account for it, then the simplest explanation, the one requiring the fewest additional assumptions, would be that something alive is producing it. Not necessarily intelligent life, not necessarily anything complex, possibly just microbial organisms floating in a planet-wide ocean, metabolizing sulfur compounds and releasing DMS as waste, much like phytoplankton do in Earth's seas. The Haitian world model made this scenario seem not only possible, but plausible. A thick hydrogen atmosphere would provide greenhouse warming, keeping the ocean below liquid even at orbital distances where a thinner atmosphere would freeze solid. The confirmed presence of methane and carbon dioxide hinted at active chemistry, possibly biological carbon cycles. The absence of ammonia fit the ocean absorption hypothesis. And now, potentially, a sulfur compound tied to life. The pieces seem to align. Madhusudan has argued that Haitian worlds could be far more common in the galaxy than Earth-like rocky planets, and crucially, far easier to study. Their larger size means they block more starlight during transits, and their thicker atmospheres produce stronger spectral signals. If life can arise in such environments, then the search for biosignatures might be more tractable than previously thought. K2-18b became the test case for that entire hypothesis, a single planet carrying the weight of a paradigm shift in astrobiology. Within days of the announcement, independent researchers began publishing their own analyses of the exact same Webb data. The results were sobering. Jake Taylor, an astrophysicist at the University of Oxford specializing in exoplanet atmospheres, reprocessed the MIRI observations and found no statistically significant evidence for DMS or DMDS. His analysis suggested the features Madhusudan's team had identified were indistinguishable from instrumental noise, random fluctuations that happened to line up, by chance, with where you'd expect a DMS signature to appear. Taylor wasn't alone. Over the following months, multiple independent groups, including researchers named Lustig-Jaeger, Wellbanks, Tsai, and others, conducted their own retrieval analyses using different modeling frameworks. Several reached the same conclusion: the supposed biosignature signal was not robust. One particularly thorough study tested the spectrum against a library of 661 different molecules, checking whether any other compounds might fit the data as well or better than DMS. The result? Yes. Molecules like ethylene and chloroethane matched the observations just as closely, sometimes even more closely than DMS or DMDS. That meant the signal, even if real, couldn't be confidently attributed to any one specific chemical. Another study dug into why different teams kept getting different answers from the same dataset. The culprit, they found, was a technical choice called binning, how you group individual data points together to smooth out noise. Depending on which binning configuration you chose, the DMS signal either appeared or disappeared. Only about 1 in 8 reasonable binning choices actually supported the detection. That's a red flag. A real signal shouldn't vanish and reappear based on arbitrary processing decisions. Then, in July 2025, a NASA-led study attempted to reconcile the conflicting results by comparing outcomes across multiple research teams using multiple methods. Their conclusion was nuanced. They confirmed, with high confidence, that K2-18b does have a water-rich atmosphere, possibly with water making up as much as half the atmosphere's total mass by volume. Methane and carbon dioxide were also robustly confirmed, but DMS, the NASA team found no conclusive evidence supporting its presence. The exciting headline claim remained unverified. This kind of scientific pushback is not a failure. It's exactly how the process is supposed to work. Bold claims get tested, data gets reanalyzed, competing explanations are explored. Certainty is built slowly through repeated confirmation, not through a single dramatic result. Strip away the controversy, and here's what we know with high confidence. K2-18b is a sub-Neptune exoplanet with a thick, water-rich atmosphere containing methane and carbon dioxide, orbiting a red dwarf star in the habitable zone 124 light-years from Earth. That alone is extraordinary. Before Webb, no telescope had ever confirmed carbon-bearing molecules in the atmosphere of any planet sitting in the temperature range where liquid water could exist. K2-18b was the first. That makes it a landmark in exoplanet science regardless of whether DMS is eventually confirmed. The planet is real, the atmosphere is complex, the chemistry is active, and the world fits, at least broadly, the profile of a Hycean planet, a theoretical category that, until recently, existed only on paper. The water detection is not tentative. Multiple independent teams agree on it. The methane and carbon dioxide are similarly robust. These findings open the door to more refined questions. What is the temperature structure of the atmosphere? How thick is it? Where does the atmosphere end and something else begin? Are there clouds, hazes, layers of different composition at different altitudes? We also now know that transmission spectroscopy with Webb works. The technique is sound. The data quality is sufficient to distinguish between different atmospheric compositions, even at interstellar distances. That capability will be applied to dozens of other exoplanets in the coming years, building a statistical sample of atmospheric chemistries across different planet types, different stars, different orbital configurations. K2-18b is the proof of concept. Additionally, the debate itself has clarified what kinds of follow-up observations are needed. More transits, longer integration times, higher signal-to-noise ratios, observations at additional wavelengths to cross-check results. The roadmap forward is clearer now than it was before the controversy began. In March 2026, a separate team took a completely different approach to studying K2-18b. Instead of analyzing starlight filtered through its atmosphere, they searched for technosignatures, evidence of technology. Using the Carl G. Jansky Very Large Array and the Meerkat Array in South Africa, researchers observed the K2-18 system across multiple sessions, covering at least one full 33-day orbit of the planet. They scanned radio frequencies from 544 megahertz up to 9.8 gigahertz, looking for any narrowband transmissions that might indicate deliberate communication or leakage from an advanced civilization. They found nothing. After filtering out terrestrial interference and accounting for the planet's orbital motion, which would cause any genuine signal from K2-18b to shift in frequency over time, no transmissions of artificial origin were detected. That doesn't rule out life, of course. It rules out the kind of technological civilization that broadcasts radio signals into space. Most life on Earth, for most of Earth's history, would have been similarly undetectable by this method. Meanwhile, the atmospheric debate continues. K2-18b remains on Webb's observing schedule. Additional transits have been and will continue to be observed, accumulating more data to push detection confidence levels higher. Some researchers have suggested that fully resolving the DMS question may require the next generation of space telescopes. NASA is in the early planning stages of a mission called the Habitable Worlds Observatory, specifically designed to directly image and characterize potentially Earth-like planets around nearby stars. That mission, however, is unlikely to launch before the mid-2030s. In the meantime, K2-18b has become a benchmark case in astrobiology. It's cited in discussions about what constitutes sufficient evidence for a biosignature claim. It's used to teach students and the public about the difference between detection and confirmation, between a signal and proof. And it serves as a cautionary tale about the gap between a preliminary scientific result and the headlines that result often generates. Researchers have noted parallels to two earlier cases. In 2020, a team announced the detection of phosphine gas in Venus's atmosphere, initially framed as a possible biosignature. Follow-up analyses identified alternative explanations and potential instrumental artifacts, and the claim remains unresolved. In 1996, scientists reported possible fossilized microbial structures in a Martian meteorite, prompting public excitement and even a presidential statement. Later work showed the mineral formations could be produced by ordinary geological processes, and scientific consensus gradually shifted away from the biological interpretation. Both episodes underscore the importance of independent verification and the danger of overclaiming based on preliminary data. So here's the number I promised you at the start, the one that breaks intuition: 8. 8 independent research teams, all analyzing the same raw data from the same telescope, and they cannot agree on whether a single molecule is present in a planet's atmosphere 124 light-years away. Not because anyone is wrong or dishonest, but because the signal, if it exists at all, sits right at the edge of what our best instruments can measure. That's not a failure of science. It's a revelation of where the frontier actually is. K2-18b will remain in the headlines. More observations are coming. The statistical confidence will either climb towards certainty or collapse into noise, and either outcome will teach us something profound. If DMS is eventually confirmed with 5 sigma confidence and no abiotic explanation is found, we'll be staring at the first real evidence that life exists beyond Earth. If the signal fades under further scrutiny, we'll have learned a hard lesson about the limits of our current technology and the ease with which we can fool ourselves into seeing what we hope to find. But even now, even without a confirmed biosignature, K2-18b has already changed the conversation. It's shown us that water-rich atmospheres exist on planets in the habitable zones of other stars. It's demonstrated that we can study their chemistry in detail. And it's proven that the search for life beyond Earth is no longer a matter of speculation, it's a matter of data, instruments, patience, and rigor. The answer is out there. We're just learning how to ask the question properly.